trying B6. The nomenclature comes from leukocytes, which are white blood cells that mediate inflammatory responses and basically are the caretakers of the immune system. This picture here is leukotriene B6 in a docked pose within the receptor PLT1, but I'll get more to that later. First I want to talk a little bit about what are leukotrienes and what kind of activities do they have and how do they respond to the system. Leukotrienes are a class of lipid mediator which feature a triene motif. And for those of you who are not organic chemists or don't take any note of organic chemistry, it's 3 conjugated triple bonds within the lipid mediator. So these are derived from basically 2 primary sources. Something called arachidonic acid, which composes about 20% of your cell life. Docosahexaenoic acid, which you might be more familiar with as fish oil, omega-3s, etc. And there's also EPA, or icosapentaenoic acid, which is more involved in resolution phase behaviors. Arachidonic acid and DHA are both enzymatically functionalized within leukocytes. And in the process of that happening, they ascertain hydroxyl groups at various positions within and upon the molecule. This changes their biochemistry significantly because suddenly you're taking a fatty acid and now you have these negatively charged, these polar groups attenuated onto it. And this is great. Suddenly you can do things with it. It's not just a fat floating around in solution. One can form a vesicle, it can bind to receptors and have unique abilities. So the bioactivity of these leukotrienes, these fatty acids that are oxygenated, are largely mediated through G-coupled protein receptors. G-coupled protein receptors are a very, very common receptor in biological systems. They are composed of 7 transmembrane alpha helices, and there's about 890 of them within the human body. They pose a rather significant challenge, however, in computational modeling. So the activity of leukotriene B4 and what I found to be the activity of leukotriene B6 is such that it is expelled into the extracellular fluid after biosynthesis. At this point, antagonists can intervene. In this case, a PLT1 antagonist can intervene. They bind to the receptor PLT1, which is the G-coupled protein receptor that I wanted. This activates neutrophils. And in the case of leukotriene B4 and leukotriene B6, this resulted in pro-inflammatory responses. And what that means is basically the neutrophils are gearing up with their chemical weapons. They're ready to start war upon the bacteria or whatever else is going on in the biological system that needs to be destroyed. What this results in is the formation of reactive oxygen species. They're very potent chemotactic elements. So if you have a leukotriene emitted in one part of the body, very rapidly neutrophils will want to swarm in on that location. And it also results in the release of lysozymes, which are enzymes that degrade biological materials. Sorry. So in order to get a grasp of PLT1, I had to produce an homology model. As there is not a wide variety of crystal structures available for use, I chose to use rhodopsin, which is a very common template protein for the construction of G-coupled protein receptors. This process of developing a homology model begins by the alignment of your target protein's amino acid sequence onto the amino acid sequence of your template receptor. So I took the amino acid sequence of BL21 and I aligned it with the amino acid sequence of rhodopsin. Now this allowed me to identify regions of space within the protein in which you would have residues that are characteristic of a certain type of structural identity. So you might have residues that are relevant to alpha helices, while they would not be relevant to, say, beta helices. And perhaps you have residues that signal, or a combination of residues that signal the beginning of a loop within the protein, an extracellular, intracellular loop of the protein. And you can identify these things and line the sequences up and get a good idea of how everything comes together And after that is performed, you have to model the loops again, because it does not make sense, and it should not make sense to me, that the loops or the extracellular intracellular loops of rhodopsin would be the same for BLT1. BLT1 has a different amino acid sequence, so of course the geometry, the spatial geometry of BLT1's loops would be different. And this is a very critical thing, because not only does it open up receptor sites and modulate the way in which the lipids and my ligands will interact with the protein, but it also tells us a little bit about more about what kind of structural changes will happen once the ligand is bound. Because the loops serve as sort of anchors. If you're pulling apart straws, you have something maybe like a paper clip that's attenuating two straws together. And as you pull them apart, you have to have some kind of friction. On top of that, I minimize the energy of the BLT1 afterwards. In this picture here, this is a projection of the energy, the force field energies calculated for BLT1. The darker the blue, the better, basically. So there's a couple of problem regions, but there's always problem regions with these kinds of things. It's never exactly perfect or fantastic anyway, but you just do the best you can. So the next step was the identification of the active site. There was a work done by Babu et al. At, I believe, Kansas State University, where they modeled LTB4 bound to BLT1. So I used their works as somewhat of a template, as they coupled it with mutagenesis data, which showed which polar residues in the active site were critical for the binding of LTB4. On top of this, I was also able to systematically go through the protein homology model that I generated and identify the receptor site by scoring various docking poses within itself. And then from there, zooming in on one location, which I summarized to be the receptor of BLT1. Excuse me. So one issue with these kind of ligands is they have like 16 to 18 rotatable bonds. And in order to identify what is the ideal conformation, you have to go through stepwise generation of the best poses within the receptor side of the protein. So I would go through and make maybe 100,000 different conformations within the receptor side, different ways in which it could sit, different geometries. And through that, I was able to figure out which ones were the lowest energy, and again, throw those away. Because it's not just the lowest energy that I want, it's the best interactions. The ligand is not just going to sit into the protein receptor site without any kind of hydrogen bonding or chemical effects that are holding it inwards. So using Glide and AutoDocFind, I was able to find the receptor site of BLT1 and characterize it. These pictures here are pictures of LTB6 bound to the receptor site of BLT1 after about 10 nanoseconds in molecular dynamics simulations. Molecular dynamics allows me to provide, it provides me with a look into the configurational space that the lipids will occupy when they're bound to BLT1. So over the course of 20 nanoseconds, I found a couple of interesting things. I was able to identify residues that were critical to the binding of these lipids to the BLT1 receptor. I was able to study how the alpha helices push outwards as the lipid explore configurational space within the protein's receptor site. And I was also able to identify, on average, which residues within the receptor site are critical to the activation of BLT1. This picture down here is the model system. So what it is there's water, there's lipids, and there's also salts. So it perfectly models biological systems. Well, not perfectly, but as close as you can reasonably assume. So I found a couple of interesting things. I found leukotriene B6 also plays the same role as leukotriene B4 in binding to arginine 156, a residue that is kind of at the top of the receptor site. And this residue is very important because the models have all shown that the leukotrienes go inward and stick in more or less from head to tail into the receptor, kind of lengthwise. Over time, however, I found something rather interesting. Leukotriene B4 did not behave as previous models had predicted. And the reason why this is these previous models had only considered 5 nanoseconds of simulation time. So they were not able to even chemically converge on the configuration of leukotriene B4 into the binding sites of the LT1. They showed a similar conformation to LTB6, which should be in that position because of the peptide bond of cysteine 97, whereas leukotriene B4 does not have the right stereochemistry for that interaction. So over time, leukotriene B4 was able to form a pin-like shape, which minimized its energy, and over time, I found the energies remain constant. Now, I'm not saying that it's entropically converged, because that would require unfolding and refolding and various other geometric conformations that were not ascertainable and will not be ascertainable unless you run these simulations for a very extended period of time. So, in order to compare this, I looked at a antagonist, leukotriene B4 dimethylamide. This molecule is not biologically synthesized. This is it essentially blocks the receptor site of BLT1, and this allows LTB4 and LTB6 to bind to the receptor site of BLT1. It does this largely mediated by the fact that the dimethylamide functionality sort of blocks off any kind of way in which the carboxylic acids LTB4 and LTB6 are binding to arginine 156. So here I have basically a graph that shows the critical 4 residues of the BLT1 receptor site. As you can see, both LTB4 and LTB6 are very involved with arginine 156, 267. Arginine 171 is very important for LTB6. And cysteine 168, this is mediated through the peptide bond, not the actual R group on that molecule. So in order to validate my theoretical model, I created a series of experiments to study how neutrophils in vitro would react to being treated with LTB4, LTB6, and a positive control orbital myristate acetate. So what this consisted of is my colleagues in the biology wing of our lab treating the mice that we have with a polyglycan called zynosoma A. This causes a severe infection of the peritoneal gland, or the peritoneal cavity of the mouse. And after that, we would harvest the neutrophils, and I would save them for LTB4, LTB6, and PMA reactive oxygen species generation. So I'm looking for the chemical weapons, essentially. With superoxide dismutase as a negative control. So what my results showed was that LTB4 and LTB6 both had a positive amount of reactive oxygen species generation in solution. They did not add linearly in terms of their activity in activating neutrophils. That is to say that I do not presume that LTB6 is interacting with a different receptor than LTB4 is. And in combination. And so this gives you an idea that leukotriene B6 does in fact have pro-inflammatory behavior on an experimental basis as well as a theoretical basis. So what does this all mean? Leukotriene B6 is classified as a pro-inflammatory lipid mediator. As a pro-inflammatory lipid mediator derived from fish oil, it conflicts with the current model of lipid mediators in which DHA or fish oil is only involved in resolution and anti-inflammatory immune modulatory signaling pathways. And BLT1 appears to serve as the endpoint signaling pathway for LTB6. Future directions include biosynthesis of LTB6, as it should follow the same pathway as LTB4, and determining the kinetics of LTB6 biosynthesis. So yeah, thank you to SERP and the Rose Hill Foundation and the rest of my lab, and for listening to me talk.
How do you measure reaction production?
So there's an enzyme called xanthine oxidase. So the reactive oxygen species almost react immediately to form hydrogen peroxides and in solution in vitro. Xanthine oxidase turns that back into the superoxide anion, and the superoxide anion reacts to forms a dye. So by oxidizing that dye, you get it to turn yellow, and then you can measure its levels.
Any other questions? Yeah, that's all pretty much in the room.